So when we look at the mechanism a little more closely, we have a club-shaped thickening at the end, the presynaptic neuron. So that's also known as an axon terminal. And then that area we have a lot of little small vesicles, little small cell membrane filled vehicles basically that harbor those chemicals. And when the action potential comes down to that area, the vesicles are released. The vesicles with that neurotransmitter in it are released into the synaptic cleft via exocytosis. So the cell membrane just glues to the cell membrane of the axon terminal and the chemicals are released to the outside. And then the neurotransmitter will bind to a receptor on the corresponding area which receives that specific chemical, and that's on the postsynaptic membrane, and then that causes either a depolarization, a stimulating impulse, or a hyperpolarization, and that's an inhibitory impulse. So that means then basically, if it's an excitatory impulse making a depolarization, that makes the recipient fire or do something. An inhibitory impulse, of course, what that does, it's inhibiting that neuron to do something. We have multiple types of synapses. It just depends, we can describe them depending on who talks to whom. So we have one synapse, it's the main one, that's an axodendritic synapse. That means an axon talks to a dendrite. We have an axosomatic synapse, and that's a synapse that axon talks directly to a soma. And then we have an axo-axonic synapse, and that is a possibility that an axon will talk to another axon and stimulate that axon directly. And that's very powerful. Synapses are often newly formed. They also disappear. And that depends on the frequency of their use. That's a very important concept, because that is what we actually do when we learn, when we create memory. So when we fire a neuron constantly, what happens is the receiving neuron will make an extension firing back to the axon of the sending neuron. And so that strengthens that pathway greatly. And so we have a saying in neurology that says neurons that fire together will wire together. That's described, when we look at that from an electrical standpoint, we call that a reverberating circuit. And that's very important. So the more you think about something, the more you the more you create a thought that's repetitive, the stronger that thought gets. You can use that for the positive, like let's try to focus on the nice sunset or the nice sunrise or a tree and be grateful and have gratitude created. Or we can learn, we can review our material and then ask us questions, so we have to expose ourselves to the material. Of course, if we space out, then that's what we do, and that gets strengthened. That's, of course, a problem. But this concept of neurons that fire together, wired together, is also in the negative. If we're always negative about everything, that thought becomes stronger. So that's a very, very important concept in not just neurology, but also psychology. I actually picked it up heavily in a book called Buddha's Brain by Rick Hanson. It's a very good book if you want to learn more about happiness and contentment. Alright, that was the synapse. Now let's briefly talk about the blood-brain barrier. The brain is in an enclosed space, and that makes it very difficult to deal with swelling and inflammation. And so we have a blood-brain barrier that regulates what molecules can go into the brain tissue and what cannot. Water-soluble substances such as carbohydrates and protein require special carriers, whereas fat-soluble substances can cross through it freely. And the blood-brain barrier is created by a special type of glial cells, and that glial cells you see here on the right, it's the astrocyte, you could see that, and that glial cell envelops the outer wall of the capillaries that go to the brain, that will leak into the brain, but those capillaries are clinging to that, those astrocytes are clinging to that capillary and enclosing it And we call those pseudopod or processes. So they're like pod refers to feet, kind. And so they seal that capillary up and make it very difficult for substances to just go through. And so those only substances now go through that we want to go through. And that's basically what the brain barrier is. Just we have to make sure we are very protective of what goes in and out of the brain, or what goes into the brain particularly. What we commonly call a nerve in our language are actually only the axons, the cables. And nerves consist of hundreds of those axons that carry impulses to the CNS in as afferent sensory nerves, or they carry impulses away from the brain and the spinal cord as efferent or motor nerves. And when we talk about mixed nerves, we talk about nerves that have cables that go in both directions in it. So we can describe the nerves as afferent or sensory nerves, efferent or motor nerves, as well as mixed nerves. And similar to muscle, when we talk about the microanatomy of muscle, we can talk about everything is wrapped. A muscle fiber is wrapped, and then muscle fibers come together and they form a bigger unit, and they're wrapped. And then the whole muscle is also wrapped. And in the nerve, that's very similar. We have a wrapping around the whole nerve, and that's very rich in collagen. Always the top wrapping around something is very, very rich in collagen and embeds it in the surrounding structure. And that's known as the perineurium. Epi means on top, and so that main layer, the perineurium, surrounds the entire nerve. And then when we look on the inside, we got multiple bundles of nerve fibers, and those are known as fasciculi. And those bundles, they're similar type nerves, and they're also wrapped, and they're wrapped in what we call a perineurium. Just wrappings, envelopes. And then the fasciculi on the inside are composed of many, many individual nerves, and those nerves together with their associated myelin sheath, that's the fat wrapping around it, they're also covered with a membrane, and that membrane is called the endoneurium. Sometimes they call that the neurilemma as well, but endoneurium is sort of a more used term. And these connective tissue coverings, what they do is they provide mechanical protection, and they also help the nerve get nutrients coming in. So they create pathways for the nutrients to come in. And we, of course, we can cut ourselves, and what happens is we can cut a nerve. And so nerves, to some extent, can regenerate after injury. The axons can regenerate after injury. Once the cell bodies of a nerve is damaged, we have a much harder time. But after injury, the part of an axon that's separated from the cell body will degenerate. But most of those nerves have this fat wrapping around it, this myelin sheath, and those are held by Schwann cells. And those do not die. Those cells are individual cells. They help. And they then create a channel and a pathway or a conduit for the axon to grow. And the axon can grow about 1 to 2 millimeters per day. That's really not much at all. But over time, that of course will make an extension. And so that's how an axon can regenerate slowly. And sometimes these nerves then they come back. When we take off a limb, for example, when you amputate though, then we got a problem because axons in that situation don't have a conduit, don't have a pathway. So what they do is they grow in all different directions, and they create what we know as an amputation neuroma. And you can only imagine how difficult that is for the brain to understand or integrate that this limb is gone, when the place where the nerve comes in the brain is still there, but there is no end to the nerve. And so that's a very difficult thing for people with amputations to deal with. So an amputation neuroma is what they get. Alright, well, thank you for paying attention and listening, and I hope you understand something. Take care.